Today, we're going to start working through a variant calling workflow. Ultimately, we're going to take raw Fastqc data and align it against a genome to identify variable sites in that genome and visualize some of those. But today, we're going to take a look at some of our data, and we are going to run a quality control check that is a fairly typical thing to do at the beginning of any DNA sequencing analysis using the program Fastqc. This is a video that's made as a companion to the data carpentry workshop, data wrangling and processing for genomics. This is not the whole workshop, and it's not meant to be. This is just a recap of how to run some of the code that goes with that workshop. So in this video, we are going to use command line tools, Fastqc, to run a quality control assessment across a range of a group of Fastqc files. And this is beginning to develop a whole workflow for handling genomic data with command line tools, specifically in this case for variant calling, but it'll be a while before we do that part. All right, to get us started, I'm not gonna make a video of this part because it's mostly just waiting, but I created a new folder called DC Workshop with a folder inside of it called data and a folder inside of that called untrimmed_Fastqc. And I CD'd change directory into that untrimmed Fastqc folder and used curl to download these 6 files. So you might, if you're familiar with the NCBI SRA, sequence read archive, you might recognize that these are SRA files, and these group into 3 samples. So we have 3 samples, each with paired end sequencing data files. So we've got SRR2589044_1 and _2. That is one sample. Forward reads _1, reverse reads _2. Often these will be written as like R1 and R2. That's the same kind of notation here. So we have 3 different samples, and we'll talk a little bit more later about what the difference is between these 3 samples. But for now, we're just going to do a assessment of the quality of these sequencing experiments. These all came as gzipped Fastqc data, so the fastqc.gz, which means that we're going to need to unzip those using the gunzip command here with an *fastqc.gz, which is going to unzip all fastqc.gz files one at a time. If maybe I should do a video about this later. If that's too slow in your scenario and you want to do it faster, there are ways to make that run in parallel if you have access to more compute resources. Using new parallel to run gunzip can be an exceptionally powerful way of getting that done. All right. But enough about the data for now. Our real goal here today is to work with Fastqc and run a quality control assessment. So let's first take a look at the Fastqc program. Assuming you have this installed correctly, and I'll post a link to the installation guide with this video. We would run Fastqc-H, and that's going to show us the help file here. If you don't see this output telling you about the Fastqc program, so Fastqc, a high throughput sequence quality control analysis tool, then that probably means that Fastqc is not quite installed right. So that would be your first troubleshooting step. This help file is going to tell us lots of useful stuff, but for our case, this is a pretty standard one. We're pretty much just going to do it as per this example. So we're going to run Fastqc, sequence file 1, sequence file 2, to however many Fastqc sequence files that we want to run. We will define an output directory using this -o flag. But otherwise, we don't really need to mess with this. The default behavior is for Fastqc data, but we could also handle BAM or SAM files. And we're not concerned here with a contaminant file or changing the way that Fastqc operates. There's options for nanopore sequencing, other behavioral flags here again. Okay, so here we have our path data untrimmed Fastqc from our current space here, and we see our 6 data files. Now let's make a space for our results to go into. So I'm going to make a directory, make dir called results, and then I'm going to cd into the results directory. Actually, I'm not going to cd in. We don't have to do that because we can define the output. All right. So we will make the directory. All right. And now we're going to set up a Fastqc command. So Fastqc, the way that we're going to pass in the names of those files is similar to what we did here with the ls command. So let's test this out. ls data untrimmed. Now we're going to do *.fastq, and that should give us. See, now it's given us the full relative path here to each of those files. So that'll be good. So we're going to take this part of this and copy that. And that is what we're going to pass into Fastqc here. Okay. And we're going to do a couple of things here to make our time a little better spent. We're going to do -o, and we're going to pass that into the results directory. -o defines the output directory. So instead of writing it wherever the data are stored, we're going to write it to a different results folder. I like that better. Keeps your data folder a little bit cleaner and makes the results easier to find. And then we're going to speed this up for video purposes a little bit using the threads flag. I'm going to use threads 2 here. It's just going to run 2 files at a time, and it'll go just a tad faster. All right, and that's pretty much it. Fastqc is super easy to use, assuming we've configured everything. Assuming we've configured everything okay. This looks good. So let's run it. All right, we'll get a little bit of output here once it loads some of the data. Yep, starting analysis, 5, 10, 15, 20% complete. See how it's doing 1 and 2 at the same time. If you have access to more compute resources, you could, of course, do more threads and run it even faster. There's no point in using more threads than you have files, though. So 6 would be our max here. Okay. One of the things I like about working with RStudio, even on a remote resource, is that it gives us direct access to our files. So instead of having to copy these files using SSH, we can just open these directly here in our web browser. Okay, so here's an example of some pretty standard Fastqc output. It's going to give us some basic statistics about the sequencing run. This is just kind of normal stuff. How many sequences were there? How many were flagged as poor quality? None in this case. They're all 150 base pairs in length. And all 50% GC, which these come from E. coli. So that's about what we would expect. Then as we scroll, we'll go through all of these things: the per base sequence quality, per tile sequence quality from the sequencing run, the per sequence quality scores, and then some analysis of the GC content and per sequence content, or per base sequence content. The length distribution, duplication levels, overrepresented sequences, and adapter content. So this is a full service analysis of our quality here. All right, so here's our quality distribution, position in read on the x-axis, Fred quality scores on the y-axis, and this is a pretty typical distribution for an Illumina sequencing experiment. Mostly high accuracy base calls, but the distribution on the lower end drops as you get towards the end of those reads. There's a little bit of degradation in the signal here, but it's not too bad. There's no reads are flagged as being particularly bad. In this one, we see kind of the same thing in the quality per tile. Again, no systematic degradation of signal here. We'll take a look at one real quick in a minute that does have some issues. Average per read quality score, about 36 for the median with a bit of a tail, but that's all fine. Our GC distribution versus the theoretical distribution. Based on our sequence data here, we've got about what we would expect. In my experience, if both of these, if the red line is a bell curve that's even kind of similar to the blue curve, you're mostly okay. You'll know if you have particularly contaminated data, if it's really out of whack here, but usually that's not the only problem. All right, our sequence length is always 150 because it's paired into Illumina data. Duplicated sequences, we don't really have anything to see here. We have, some sequences are duplicated maybe twice, maybe 3 times, but not much. If we had a big spike anywhere out here past maybe 6 or 7 duplications, that would be cause for concern, and that would be highlighted in the overrepresented sequences tab, but we don't have anything here for this one. We do have a bit of adapter content that we will have to deal with next time in our next session. All right, let's go look at one more. Okay, yeah, this one's a bit different. At a glance, we see several red X's over here, which draw our attention to some issues with this, some possible issues with this sequencing run. So let's go to the per base quality. Yeah, that's part of it. There's a lot of degradation in the quality scores for this sequencing experiment. After about 60 or 70 nucleotides, we have some reads at least that are fairly low quality on that end. And we can see that some of that's systematic degradation in certain tiles from the sequencing run. So some of the parts of these this read output was very low quality. So we'll see what we can make of this still. The average per read quality isn't too bad. It's just a longer tail than what we had previously. So it's really just some of these reads show a bit of degradation towards the end of those sequences. So we will have to probably do some trimming of low quality sequences here with that second file that we looked at, but we will be all right. Okay, so if you've done this exercise, go through and look at all of the HTML files, not just 2, but I'm not gonna keep recording for that. I'm gonna go sift through a bit more data here and see what we have to work with coming up.